Okay, this is lecture 16 for microbiology. What we're going to start talking about is the immune defense system. Last time we talked about mechanisms of pathogenicity. Now we'll talk about mechanisms of defense. Now, there are two major systems or mechanisms that act as our defense. One is the non-specific immune system or non-specific defense mechanism. And the non-specific immune system has been called it is now called the innate immune system or innate. So we're talking about innate immunity here. This is a built-in, already active system. Innate means inborn, native. So this is the part of the immune defense mechanism or non-specific defense mechanisms that are already present. The other one that we'll talk about in the next lecture is the specific defense mechanisms of the specific immune system, which is called the adaptive immune system or adaptive immunity. Okay, but now we're on innate immunity. So when we did speak about mechanisms of pathogenicity, we said that in order for an organism to be pathogenic, it would have to be able to carry out a certain it would have to have certain abilities characteristic of it. It would need a portal of entry. And for an organism to get in, it has to get through one of these portals of entry. Depending on which pathogenic microorganism you're looking at, they have to enter past these. And these form barriers as part of the innate mechanisms of defense. And once through, it has to be able to evade these the host defenses. Now, in terms of defense mechanisms of the host, in this case we're talking about humans, there are certain terms, there's terminology that you need to understand and be comfortable with. Resistance refers to the ability to resist an infection, to ward off disease, to not become infected. Susceptibility is the lack of resistance to a disease. If a person is susceptible to a particular disease, it's because they don't have much, if any, resistance. Yes, they have a certain amount of non-specific resistance, but for many organisms, they do get into you quite often, but not in significant enough numbers, or your own mechanisms of defense are strong enough to just ward that off. You have resistance. If you're susceptible, you don't. As I told you, there are two types of immunity, two main groupings of mechanisms. One is innate immunity, which is non-specific. So we're talking about non-specific immune mechanisms or non-specific resistance to disease. These are general defense mechanisms that are effective against all pathogens or any large group of pathogens. And then there's the adaptive immune system or adaptive immunity, which is very specific. It takes some time to develop. This is what we mean by when we talk about a person being immune, because you're specifically immune to a certain type of disease process. So you develop specific resistance to a specific pathogen if you encounter that pathogen and get infected and recover. Then you will have a specific immunity. Your adaptive immune system responds. Quite often, the innate immune system responds so effectively and quickly that the adaptive immune system never actually gets activated. So you may not even be aware that you did have some pathogens entering into you. So again, we're going to talk about innate immunity, non-specific defenses, and then later, today, in this lecture at least, and then later adaptive immunity. So there are, one way of looking at it is that we have several lines of defense, and the first two lines of defense are part of that innate immunity or innate immune defense mechanisms. The first line of defense is our intact skin or our intact mucous membranes. So we have an intact stratified squamous epithelium, and also we have a intact mucosal epithelium on our inner surfaces that are both in contact with the external environment and do come into contact with potential pathogens. The other important aspect or part of the first line of defense is the normal microbiota that populate our epithelial surfaces and act to compete against any pathogen trying to gain a foothold, or compete against the few that are present so that they never gain an advantage and start an infection. The second line of defense, and we're going to talk about all of these in more detail in the next few slides, but the second line of defense, also part of the innate immune system or innate defense mechanisms, are the phagocytic white blood cells. These cells, phagocytic white cells, include macrophage monocytes. Monocytes, when they're in the bloodstream, when they leave the bloodstream into the tissues, they're called macrophages. So the general term is monocyte/macrophages. I'm talking about the same type of cell there, one type of cell, a large family of cells. Maybe that's a better description. So we have the macrophage monocytes and, of course, the neutrophils. Now, all the neutrophils are granulocytes, and all the other all granulocytes, basophils, neutrophils, and eosinophils are in fact phagocytic, but it's mainly neutrophils that are the main phagocytic cell, those and macrophage monocytes. So we have, again, macrophage monocytes and neutrophils as the main phagocytic white blood cells. That's an important part of the second line of defense, and they mediate in part a very important process called inflammation. The inflammatory response is a very, very powerful defense mechanism. Another aspect of the second line of defense is fever, and another one are antimicrobial substances. These are small proteins called peptides. Small proteins in general are called peptides. These are small peptides that have direct antimicrobial effects on organisms, pathogenic organisms. They are, for the most part, proteins that you produce. It's part of your normal secretion of the mucosal membranes, and they play an important role as part of the second line of defense. So phagocytic cells, macrophage monocytes and neutrophils, the inflammatory response, fever, and antimicrobial peptides or proteins or substances. Later we'll talk about the specific immune response, which forms the third line of defense. Not today. Not in this lecture. So let's talk about the first line. The first line, as I said, is the intact skin here, or mucous membrane. Here we'll talk about the skin. It's the stratified squamous epithelium. In this photomicrograph, what you see is the epidermis here. And remember that some areas of epidermis have a thicker layer on the outside here, all this, like the bottom of your feet, for example, or probably on, where you've built up some callus, thicker layer. This is all dead cells, keratin, filled with keratin. Keratin is a protein. As these cells are produced, and they are, of course, produced, just a second. The cells of the epidermis are produced in the lower layer here at the base of the epidermis, right between the junction of the epidermis and the dermis. This is where the stem cells are, and the cells are produced and then get pushed upwards. And finally, I'll put an arrow in a different color. They get pushed up as they accumulate. They're produced, and so they're constantly getting pushed up, and you're producing, it's not very dark, is it? They're constantly being produced and pushed upwards as the more are produced in the lower level. And then they flatten out and they die, and you can build up actually quite a thick layer of dead squamous epithelial cells. And what that dead material is mainly a protein called keratin, which are in these cells and is all that remains, and is quite a resistant material. It forms quite a good barrier to the outside. So we have the stratified squamous epithelium of the epidermis. The top layer is dead and constantly gets sloughed off, and a new layer gets produced and pushed up from underneath, and that protects you from the outside world. As I said, the top layer is composed mostly of keratin, a water-insoluble protein that forms quite a nice barrier. Immediately under the skin is the dermis. That also has some protective effect, but mostly it's this buildup of skin. In the dermis, by the way, is down here. It's this layer down here, from here to here, from here to here, right? So all of that area is dermis. Above that is epidermis, and the very top layer is our dead keratinocytes, or epidermal cells that have died. A nice barrier in your skin. On your inner surfaces, mucosal epithelium, again separating you from the outside world. Mucosal membranes, or mucosal epithelium. They line all of your body cavities, GI, GU, respiratory. They protect you. They're lining your gut, your lungs, your genitourinary tract. These cells produce mucin and secrete it as mucus and acts as a lubricant. In the respiratory tract, you have the ciliary escalator mechanism, which constantly particles, and including microorganisms, get trapped in the mucus of the respiratory tract as you breathe it in. They stick to that, and then they get pushed up by the synchronous beating of cilia. The cilia, of course, are on the apical side of the pseudo-stratified columnar epithelial cells that line your respiratory tract. The lacrimal apparatus, this is the apparatus, the structures that produce tears. They wash away particles that get into your eye. Saliva also is part of the mechanism of protection associated with mucous membranes, regularly washing away microbes that might accumulate in the teeth and in the mouth. Urine has an effect. Urinary tract, as part of the genitourinary tract, is lined with mucous membrane, and the urine flows and carries microbes away. So the flushing action of urine is important in terms of protection against pathogenic organisms. Vaginal secretions, the mucosal secretion of the lining the vagina, also helps remove any microbes and menstrual tissue that are present. So those act as important barriers. Here we look at the lacrimal apparatus. It's not so big that you see, as shown in this diagram, I would say, but it does explain. Here is these are the glands that secrete tears. They're in the on the lateral superior side of the housing of the eye, the orbital cavity, and they get released, and they flush across the eye and move things out. The eye itself is covered with a thin, clear epithelium, and they drain down the lacrimal canal, and the lacrimal canal drains into the nose, which explains why, when you cry, your nose runs. Partly, at least. That's because of tears draining down. Here's the mucociliary elevator mechanism, or escalator mechanism, I'm sorry. In the respiratory tract, it's a high-powered electron micrograph using electron microscope. And here's a nice diagram of the pseudo-stratified columnar epithelium. There's the thickness of it. So this is it's the apical surface on this side. There's the basal surface on that side. You have immediately below every epithelium, you have a basement membrane. That's this thin layer of secreted connective tissue, the basement membrane, right? At the here, these are goblet cells. These represent goblet cells here and here and here, producing mucin, which they secrete. And when it combines with water, it becomes mucus. You also see the cilia that are, and you see it very nicely actually in the electron micrograph. These cilia are constantly beating upwards. They slowly drop down and then beat forwards, upwards to move that mucus and trapped particles with potentially some microorganisms in it out of the respiratory tract. Okay, let's talk about some of the chemical compounds or factors that are produced that assist in nonspecific defense mechanism or the innate immunity, part of the all part of the first line of defense. I'm not sure if this is the first or the second line of defense. Let's go with first for now. The skin or the skin or the stratified squamous epithelium has sebaceous glands in it. These secrete an oily substance called sebum. It has significant fungostatic fatty acids in it. The fatty acids in it have fungostatic activity. They prevent fungal spores from growing. And it prevents the skin from drying out and cracking. A crack, of course, would, give an entry point. The fatty acid and also some lactic acid produced by the skin gives a fairly low pH, and that also helps to inhibit microbial growth. The other thing is that your secretions like perspiration from sweat glands, tears, saliva, and some of your other tissue fluids that are excreted out contain a protein called lysozyme, which has a powerful antibacterial effect. It is capable of breaking down peptidoglycan, which forms an important part of the cell wall of bacteria, and therefore can kill bacteria in that way. So all of these contribute to nonspecific defense. Gastric juice, of course, also does. The gastric juice is the secretions from the gastric glands. Hydrochloric acid, for example, is very low pH and is quite effective, in fact, at killing microorganisms. In your blood, you have proteins that bind up iron and make it unavailable to any microorganism that could get into your blood. Every living thing needs iron, and in your bloodstream, in the plasma, you have transferrin that binds to iron very tightly, very firmly binds to any free iron, making it unavailable. There is also a gas that gets produced and dissolved in the blood, which is called nitric oxide, and it can inhibit energy production by microorganisms and DNA synthesis. Their energy production, ATP production, and the DNA synthesis. That occurs mostly with inflammatory responses, mostly as a product of macrophages. So all of this is the first line of defense that I'm talking about. And the last thing are the normal microbiota that have an antagonistic or competitive effect on any pathogens that might be present or you might get contaminated with on your outer or inner surfaces. Okay, let's talk about the second line of defense. Again, it is part of the innate immune system. It is nonspecific. So we'll talk about phagocytic cells, inflammatory response, fever, and antimicrobial molecules. Actually, I realize now I made a mistake when I was introducing the idea of antimicrobial molecules. I should have really talked about small proteins like lysosome as part of the first line of defense, which I did eventually, but for second line of defense, molecules that have antimicrobial effects, it's more specific. We're talking about things like complement, and I'll talk about that now. Okay, so when you look at the white blood cells in general, you have about 75%, the vast majority of white blood cells are in fact granulocytes or polymorphonuclear cells, PMNs. And as you know, granulocytes there are three types: neutrophils, basophils, and eosinophils. Most common being neutrophils. So anywhere from 2/3 to 3/4 of the white blood cells are granulocytes, and most of those are in fact neutrophils. You have a few basophils and some eosinophils. You also have monocytes. On average, say about 5% roughly of the white blood cells are monocytes. And you have lymphocytes. The lymphocytes form 20-25% of the cells. Lymphocytes are part of the adaptive immune system, the specific adaptive immune system. They will not be discussed much here. They're part of that third line of defense, or specific adaptive immune system. Okay, the white blood cells in the body, in terms of which are called leukocytes, in terms of the phagocytic cells, include the granulocytes and the macrophage monocytes. Macrophage monocytes, these are cells in your bloodstream, monocytes, and when they leave the bloodstream and go into the tissues, they are called macrophages. The granulocytes, there are three types: basophil, neutrophil, and eosinophil, but it's mainly the neutrophil that has the phagocytic activity. So most of the phagocytic activity among the granulocytes is by neutrophils, and then there's also the macrophage monocytes. These, of course, are all myeloid lineage cells. These cells, lymphocytes, are part of the adaptive immune system that we'll discuss later. There are a number of different cells in the blood, of course, but we're just going to be talking about the white cells that are part of the innate immune system. So we have the granulocytes, neutrophils, basophils, and eosinophils. And these, of course, are characterized by the polymorphonuclear structure of their nucleus, the multiple shaped structure of their nucleus, and the fact that they have granules in their cytoplasm, and the granules stain differently. Eosinophils, they're red. The granules are red staining. In basophils, these cells are purple. The granules are purple staining. And in the neutrophils, they're very lightly lilac staining, or not stained at all, hardly. During a infection, a typical initial sign of an infection is an increase in the total number of white blood cells in the peripheral blood, and this is termed leukocytosis. So if a person has leukocytosis, that's an indicator, a suggestive of, not proof, but suggestive of infection. The neutrophils are mainly phagocytic and will take in any microorganism, foreign microorganisms that get into the body. They will not. They will move towards the area where where the microorganisms are present, and they'll leave the bloodstream, move towards that area. They'll engulf them, take them in by phagocytosis, and kill them by quite potent mechanisms. Basophils, on the other hand, although they are somewhat phagocytic, mostly their response to, and it's a little more complicated, we'll talk about it later, they will degranulate if triggered appropriately, and their degranulation, their granules contain quite a bit of histamine, and this causes increased vascular permeability and increased edema in the area. They contribute towards a antibacterial effect. Again, we'll talk about it mostly later. Eosinophils are, in fact, active against mainly parasitic worm infections, although they do have some phagocytosis activity. Monocyte macrophages are phagocytic. The degree of phagocytosis, or the phagocytic ability, does increase tremendously when they leave the bloodstream and mature into macrophages, but they do have their own somewhat phagocytic activity. There are macrophages present naturally in the different tissues. Most tissues in the body have their resident macrophage population that came from monocytes that left the bloodstream during development and entered and lived there permanently. You have alveolar macrophages in the lungs. You have Kupffer cells in the liver. You have bronchial macrophages. You have macrophages in the brain that are called microglial cells. You have macrophages in the skin that are called dendritic cells. All of these are resident macrophages or tissue macrophages. They did not move into that tissue because of an inflammatory reaction. They are already there and can respond to entry of microorganisms. Phagocytosis essentially is ingestion by engulfment and taking in of a foreign particle or microorganism by the cell, the phagocytic cell. It envelops it and surrounds it with membrane and takes that in and has it in its cytoplasm as a vacuole, a membrane-bound vacuole. Phagocytosis breaks down the origin of the word is from the term in Latin to eat and cell, so it's a cell that eats. Here you see the progression of events that occur with phagocytosis. Presence of a microorganism will trigger accumulation of phagocytic cells to the site. They will, which is that movement towards the site of infection, is called chemotaxis. They'll leave the bloodstream and move towards that site of infection. The first cells typically that will arrive are neutrophils, followed soon after by macrophages. And they can both phagocytose, and you see that here in 2, formation of a phagosome around, which is essentially a vacuole, or phagocytic vesicle, or membrane-bound vacuole, called a phagosome, around the ingested organism or particle. And then other vacuoles that contain hydrolytic digestive enzymes will fuse with the phagosome. These are called lysosomes, and they will fuse with the phagosome to form what's called a phagolysosome. And now the digestive hydrolytic enzymes can act on whatever it is that was ingested into the phagosome. You know, by enveloping these enzymes within a membrane as a separate compartment in the cell protects itself from their action, so they don't act on the cell itself, and only with fusion will act on the thing that is foreign to the body inside the phagolysosome. And then you get digestion, it breaks it down and helps to kill it. There are other mechanisms operating besides digestive enzymes. And anything that can't be broken down gets ejected out of the cell, discharged out of the cell. Here you see an electron micrograph of a macrophage and some bacteria. You can see these rod-shaped bacteria quite nicely here and here. All of these are rod-shaped bacteria, you see, and the cell is moving outwards towards them, and it's going to engulf them. All of this is the macrophage here, all that. Again, a macrophage engulfing, in this case, the bacteria are Neisseria gonorrhea, the organism that causes the sexually transmitted infection, gonorrhea. Neisseria, the genus Neisseria is characterized by arrangement of pairs of bacteria. When you see round bacteria in pairs, here's another two there. That's a suggestion certainly that there's two there. Suggestion, these are all in pairs, you see. Certainly that's the way Neisseria grows and arranges itself. So here you see another case of a phagocytic cell being attracted towards these bacteria. There are many mechanisms that microorganisms use to try to evade phagocytosis. One of them you learned about, which is having a capsule, because the capsule has an effect on phagocytosis. It'll inhibit phagocytosis. Streptococcus pneumonia certainly does produce a capsule, and that will lessen the ability of phagocytic cells to adhere to and engulf, phagocytose the organism. There are all kinds of things that different bacteria have evolved, and some viruses have evolved to avoid phagocytic defense mechanisms. Now, let's move on and talk about the process or mechanism of inflammation, the inflammatory response. It's an extremely important response that helps defend you. It is part of the innate immune system or nonspecific immune system, part of the second line of defense. It was described about approximately 2,000 years ago. And there are what are called four cardinal signs of inflammation. They include the following: heat, redness, swelling or edema, and pain. Those are the four key characteristic signs of the inflammatory response. And they were first described, written about in Latin. And the Latin for those terms, I think, is worth knowing. And you can, if you know any Latin-based language, such as French, for example, and actually, at least in part, English, you would recognize that these are important form parts of words that are used in English, French, certainly Spanish, Italian. Calor, which means heat. This is Latin. If you learn any words in Latin, you should learn these four. Calor for heat, rubor for redness, tumor for swelling, and dolor for pain. Calor, rubor, tumor, dolor. Heat, redness, swelling, and pain. The four cardinal signs of inflammatory response. Well, inflammation has quite a bit of mechanisms involved. It's not some sort of simple, increase in heat, redness, pain, and swelling. There's lots of things involved in fact, in it. And it gets activated because of what are called acute phase proteins. They get activated. You get vasodilation, an increase or widening of the blood vessels in that area so that more blood flows into that area. And you also, besides vasodilation, you also get increased vascular permeability in the area. Now, when you have vasodilation, and I'm going to write it, yeah, I guess I'll write it on the side. Well, when you have increase in vasodilation, so there's more flow into a particular area of the body, then what you get is more blood flow into an area, so then the area will turn, in fact, red. Okay, so that's one reason why you get, or that's a reason, the vasodilation is the reason why you have redness, one of the signs of the inflammatory response. Also, blood is slightly warmer than the rest of the body, so it feels hot. So if you just think of it in terms of, say you have an area of inflammation on your arm, it will be red and it will be hot. And it's because of the increased vasodilation in the area. And the accompanying increase in vascular permeability means that more fluid will leak out of the blood vessels in the area, and therefore you get the third sign, which is swelling, edema in an area of inflammation. Okay, so we have rubor, calor, tumor, and the pain is caused by the effect of some of these molecules that are being released. When we look at all of these things, I can tell you certainly that histamine is an important part of the swelling and vasodilation that you see with the inflammatory response. And histamine is produced from degranulated basophils. Degranulation of the basophils will cause histamine release. There are other molecules, kinins, prostaglandins, leukotrienes, that can mediate some of these. You know, in an area of inflammatory disease, of an inflammatory response, the white blood cells that are flowing in the blood in that area, not only are those blood vessels swollen and you have more blood flow, and they have increased permeability so that more fluids can move out into the tissue, but also cells find it'll be easier for cells to move out. Now, for white blood cells to do that, they have to marginate. They have to get to the edge, the outer edge of the capillary. So margination is an important part of the mechanism of the inflammatory response. It allows, you know, because the cells marginate, the white blood cells will go to the margin, and then they will emigrate out of the capillaries, and you get accumulation of inflammatory cells like neutrophils or macrophages. Eventually you get, you know, normally the inflammatory response resolves the infection, and then you always, the end of the inflammatory response, ends up with, if it ends well, then you get tissue repair. So the kinds of things that these molecules that release and mediate some of these acute phase molecules that mediate a lot of the inflammatory response, including histamines, kinins, prostaglandins, and leukotrienes, they get released also by damaged cells, histamine by certainly basophils, and they cause these responses of increased vasodilation, increased permeability, especially these two, histamine and kinins. I think that's important to know. And vasodilation, increased permeability, is the cause of the redness and the heat and the swelling. These can increase, the prostaglandins can increase the effects of these. Leukotrienes can increase vascular permeability. These molecules, there are many, many different types of prostaglandins that can be produced in the body, many different kinds of leukotrienes. They are major mediators of the inflammatory response, and any of the non-specific anti-inflammatory drugs that you're familiar with, that you buy over the counter, are all directed typically at one or more of the prostaglandins and leukotrienes in your body. Part of the inflammatory response is to injury and entry of bacteria is shown here. Here you have an injury. This is a parenteral route of entry through piercing in the first line of defense, the stratified squamous epithelium, down into the dermal layer, and introduction of bacteria with that. Bacteria can come with the piercing or, enter after through the hole that penetrates it. You get release from injured cells of histamine, kinins, prostaglandins, and leukotrienes, and you start getting increased vascular permeability, as is shown here by the fact that these blood vessels shown here are thicker than up here, right? They get wider. So you get an increase in vasodilation occurs, and increased permeability occurs. So there is some swelling, as you can see. The tissue gets swollen. White blood cells, if there's wherever you have injury, you will have bleeding, so you get some clot formation. The white blood cells that marginate and move out of the capillaries towards this injured tissue, towards the presence of these chemical compounds released with injury of tissue, they will keep moving into that area and will mediate phagocytosis and will produce some quite potent molecules that can kill the microorganisms. And as long as there are still microorganisms there, more will move towards it and accumulate. They produce such toxic molecules that they not only kill the bacteria or microorganism present, but they also kill themselves. This is a way of limiting the inflammatory response. You cannot have an ongoing continuous inflammatory response with more and more white cells arriving and producing toxic compounds, because that would injure the host's tissue too much. So they have their action, and they have a limited lifespan once they're present there. And if there's still more microorganisms present because the organism's growing and multiplying, then more new white blood cells will arrive, and you'll get an accumulation of white cells in the area. So you can get an accumulation of the cells, dead white blood cells, and those dead white blood cells, of course, are pus. They form what is the pus, accumulating at a site of infection and inflammation. Eventually it does resolve, and you get wound repair. This action, by the way, of marginating and then leaving the capillaries is called, this process has a name. It is called, I'll write it here, diapedesis. So white blood cells leaving the circulation and going into the tissue, they do so by the process of diapedesis. Diapedesis. I'm misspelling it, I believe. Well, we'll see. Diapedesis. That's an S. I don't know why I was drawing an A. Just 1 second. That should be an S. Yeah, that's it, diapedesis. So they move out of the bloodstream, they go towards an area where there's an accumulation of bacteria or microorganisms, and the cells that move initially are neutrophils, followed soon after by macrophages, and they can phagocytose, so they emigrate by they marginate. They go to the edge, the margin of the blood vessel. They'll stick to the endothelium. They will emigrate by diapedesis. By squeezing in between the endothelial cells, they'll move towards the area of infection where the bacteria are, and will phagocytose them and kill themselves also. And they will keep arriving as long as there are more bacteria there. But when all of the bacteria have been killed off, then you'll get wound healing and regeneration of the epidermis by cells that replicate, and it goes back to a normal situation. That's tissue repair. Okay. I think this is a good place to stop. This will be lecture 16, part 1.